My name is Hersh Bhargava, and I'm a second-year PhD student in biophysics at UCSF. Today, I'd like to share with you research that I've been conducting to develop and validate strategies for computer-guided design of living cell-based therapeutics. For thousands of years, humans have been modulating their own biology using small molecules. More recently, biologics, that is drugs based on biological macromolecules such as antibodies, have entered the fray as a new generation of therapeutic modulators. However, if we step back and ask the question, what would our dream modulator of biology look like? I would suggest that we would land somewhere near science fiction's nanorobots. These micron-scale devices would be able to sense their environment within the body, migrate to the site where they're needed and deliver payloads like drugs, process information, and perhaps even kill or reprogram target cells. While we are a long ways off from accomplishing this kind of technology through electronic or mechanical devices, our own living cells inside of our bodies accomplish many of these functions on a daily basis. Immune cells in particular are autonomous cells within the body that have the structural basis for a number of these functions already within them. This reality has been the basis for bringing in engineered human cells as the next class of therapeutic modulator of biology. The current example of this is chimeric antigen receptor T cells, or CAR T cells, which involve expressing in a T cell scaffold a special antigen receptor that redirects the T cell's killing and activation functions against a cancer antigen. This technology has been used to develop next generation cutting edge therapies for blood cancers, which are currently on the market and have saved the lives of patients with certain diseases. As we have developed live cell therapies, more molecules have come into our molecular toolkit for engineering cells with new therapeutic functions. For example, synthetic Notch receptors are modular proteins that allow us to create sense and respond programs, such as this one, which enables CAR T cells to detect the presence of two antigens on target cells and only kill target cells which express both antigens, something that we would have a lot of difficulty accomplishing with conventional therapeutics. The motivation for my work is to look holistically at the entire space of synthetic and naturally occurring molecules which we can leverage to engineer the next generation of designer cells. These include molecules that can function as tissue GPSs, in cell-cell communication, as clamps, as locator beacons, distress beacons, and even antibodies themselves, which we can deliver using immune cell scaffolds through secretion. The question of how to engineer cells and how to draw from this toolkit to build a therapeutic function is a difficult one. Current approaches for solving this problem are largely ad hoc and require many iterations of expensive and time-consuming iteration in the lab. I propose to leverage the power of computation to guide our design decision-making when it comes to engineering immune cell-derived therapeutics. I envision a workflow in which we can search in a massively parallel fashion in silico the design space of all possible cell therapies, test some of our predictions in the lab using in vitro assays, and then validate them in preclinical and clinical models, and have a much faster and more precise cycle of design iteration. Toward this end, I've developed an open source computational modeling platform for cell therapies called CellTX, which translates biological designs into mathematical models and enables their simulation. This tool is currently available as an open source project on GitHub. Following iteration and search in CellTX and similar modeling platforms, we can validate our candidate cell therapies in vitro assays like this one under the microscope, and then move into mouse models of specific diseases where we can test their efficacy with preclinical endpoints. I hope that this work can yield a next generation approach, not only for improving the design of current cell therapies, but also open the way for next generation and novel applications of cell therapies, such as in autoimmunity, degeneration, and beyond. With that, I'd like to thank my advisors, Hannah El-Samad and Wendell Lim, my labs, the UCSF Biophysics Graduate Program, and the ARCS Foundation for supporting this work. Thank you very much.